Yeah, hello everyone. Can we have the next slide? Oh, just do this one? Yep, thanks. Okay, I'm David Booth. My lab is trying to help find a cure for MS using genetics. We know that MS is a disease which has a genetic and an environmental component, and that if you can identify the genes which are involved, that would surely help in finding a cure. So there's some very good news on that front, some milestone achievements as a result of giant collaborations and new technologies. So in 2007 we knew just one gene which was affecting MS. Now, as a result of this collaboration, which is groups from Cambridge, from all over the UK, from Europe, from the USA, from Australia, from New Zealand, they all got together and did this massive experiment which involved DNA from 10,000 people with MS, DNA from 10,000 healthy controls, and to find the genetic variants which were different. And so this experiment had enough, at last, power to answer the question. And so the results are going to be published in about released in about a month. I can tell you now that we've gone from 1 to 57 genes which affect MS. As a result of this experiment. I should say this experiment was very expensive. It was funded largely by the Wellcome Trust, but then every group who contributed to it also needed funding. In our case, we were funded by MSRA, NHMRC, and it was a big collaboration, the ANSGene collaboration, which contributed Australia's component to this. We were very lucky with the first gene. Just knowing the genes, knowing we now have 57 genes, what are they going to tell us? Potentially there are gold nuggets there, but you've got to work out why they're associated with MS, why they're affecting MS. And it's not always going to be easy. The first gene, it was easy. That was a gene called HLA-DRB1. And if I can just highlight it. Because that gene has a great trait, it's only expressed on one type of cell, and it only does one thing, which it activates CD4 T cells. So from knowing that gene was associated with MS, you knew straight away CD4 T cells affect MS. And so you could target CD4 T cells as a rational approach to coming up with a therapy. And in fact, see that little peptide there that's captured by the gene and presented to T cells, that's actually the basis for one of the drugs, glutarimer acetate, which is designed to be that very peptide. And then the fact that it shows the importance of CD4 T cells in immunopathogenesis also supported all the clinical trials that needed to be done on finding other ways to modulate T cells. So that's the interferon betas and the Tysabri. Tysabri stops CD4 T cells crossing the blood-brain barrier. It all fits into a common picture. But then looking at the 56 other genes, we don't yet know. I hope that doesn't look too complicated. This is the dominant paradigm as of 2005, and it hasn't changed. And the idea is that these cells which express that gene DRB1 activate the T cells cross the blood-brain barrier, and they orchestrate an autoimmune attack on the myelin sheath of the neurons. That's the dominant paradigm. And then what did the genes tell us? Well, the genes tell us that, as it happens, all 57 of those genes, all of those genes where we know where they're produced and we know what their function are, they're all immune, 100%. So it's looking like this is the part which is what the genetic susceptibility is about. Doesn't mean that pathogenesis doesn't involve the neuronal component, because obviously it does. But the genetic variation is due to the immune response. Specifically, what does it tell us about the immune response that we can then use to devise new therapies? Answer at the moment, it actually tells us quite a few things, because we know that those genes are largely about T cell differentiation, particular types of T cells. Here are the usual suspects. There are a number of T cell subsets, and as a result of knowing these genes, we'll be able to work out which of these T cell subsets are most important in MS, because they're the ones that will express the genes which are associated with MS, and where the genes will have their effect. And so research now will be targeting particularly that one and that one, because there's all sorts of other evidence that those are prime culprits in MS pathogenesis. But we have to do the work now to find out why these 57 genes affect these T cells. And I'll just, it's not all about genetic susceptibility. You can use genes in other ways. And this is my last complicated slide, my last slide. What we see there, this is the idea of using markers. Genetic markers or gene expression markers can hopefully tell us who might respond to what drug and are they responding to the drug, so clinically useful. This was an experiment we did as part of the ANSGene collaboration, which is funded by MSRA and an ARC linkage project. What you're seeing there is every column is an individual. So we've got a bunch of controls and a bunch of people with MS, and every row is a gene. And so there's about 200 genes that you see there. And when you see red, that means it's overexpressed, and when you see green, that means it's underexpressed. And you can see there's this signature for MS where these genes are all overexpressed and these genes are underexpressed. These genes are mainly activating T cells, so this fits with the genetic information. And we're hoping that we'll be able to use this so that you'd expect this reds turn to green on therapy, for example. So we have to do that experiment to see if that truly is the case. But you can also see that some people with MS are green and some people are red, and some people who are controls are red there and some are green. So some people have the signature anyway, which is presumably susceptibility to autoimmune diseases, which these people have, but they haven't got a disease. A lot of work to do there to make that useful clinically. So I'll just wrap up by saying that we've got 57 genes. We've got to find out why they're causing MS. And we've got other things to do with genes to try and find out how to use them clinically.